invented in 2012, was used to improve crop plants quality, and I will leave these older techniques behind. So CRISPR-Cas genome editing is a powerful tool with potential for crop improvement. It can improve yield and quality. It can improve abiotic stress response. It can also improve biotic responses of plants. And now I divided the lecture into several categories for this application of gene editing in trait improvement. It's okay? In trait improvement in plants. So first I will talk about increasing agricultural productivity, some examples. Then I will move to enhancing nutritional value of crops. Then I will move to developing climate-resilient crops. Then I will move to creating diseases and plant and pest resistance crops. Then I will talk a little bit about ethics and regulations in plant genome editing in different countries. And I will finish talking about future directions in plant gene editing, challenges, and opportunities. So we will start with increasing agricultural productivity. So it can be done, it is, by the way, I just want to ask you, do you want our presentations? Okay, so we will prepare all presentations that you can read and follow everything in more detail, right? Because not everything is visible, so it would be better than that you have it, yeah? Because, for instance, in this table that you are not able to read, in this table I provided examples of plants and pathways that were affected in order to get yield improvement. And what is in the black frame, these three examples in wheat, rice, and barley, I will present you in details. But here you have all citations for all papers I found interesting that you can later on also study if you would like to. So the first example deals in wheat with a gene that is called RPK1, and it means receptor-like protein kinase 1. RPK1, receptor-like protein kinase 1. And the enzyme is known to negatively regulate root system architecture, RSA, root system architecture. So it becomes short, and when it, how does it occur? It occurs by influencing polar auxin transport. But the growth shortage results that plants are not able to withstand well drought, and also where nutrients are in deficiency, they cannot also reach for nutrients that are deeper in the soil. So the study used CRISPR-Cas9 to edit RPK1 gene in wheat, and it led to enhanced root system architecture and higher grain yield. So you see some genes in some circumstances operate disadvantages for plant, in a disadvantage way for plant. Sometimes it is a trade-off. Sometimes you get something better, something worse, because you don't have genes that operate only in a bad way, right? But in the case of crops, you have to choose what is better to get a better yield and to produce food. So, so a part of the enhanced root system architecture, they got higher grain yield, probably because plant was able to reach deeper layers in soil for water and nutrient deposits. So mutations improved root traits. So roots were longer with increased surface area. And agronomic traits, like what we call is 1,000 grain weight. We compare 1,000 grain weight of different crop varieties, and if it is increased, it means that the efficiency of grain production is better. So in all edited lines compared to wild-type plants, the 1,000 grain weight was improved. And also, there was a higher number of tillers, leading to higher overall grain yield. There was, however, shorter spike lengths, but the grains were heavier, and that somehow compensated to the shorter spike. So these findings suggest that targeted genome editing of this RPK1 gene can enhance wheat productivity and drought resistance. So this is the first example for yield improvement. And the second one is connected to rice and deals with the gene called grain size on chromosome 2. And this is a gene that plays a crucial but positive role in determining rice grain size. Also is known as GRF4, growth regulator factor 4. Maybe you remember that the GRFs are targeted by microRNA 396 from, I think, 2 days ago, or yesterday maybe I was talking also about it. So what is the gene? Of course, this is a transcription factor that positively regulates grain development, nitrogen use efficiency, and plant growth. And the study targeted microRNA 396. That was, of course, downregulated the level of this GS2 or GRF4 gene expression. And mutant allele that targeted the recognition site in mRNA of this gene was generated, and it had some deletions, some substitutions, and it impaired a repression by MIR396 and increased GS2 expression. So if a gene is regulated by microRNA, we can also play with target site to, for instance, get out of the control of microRNA. And for that, here CRISPR-Cas was used. So what happened? So grain lengths increased by more than 21%, widths by more than 13%, and the length-width ratio was improved. Also, 1,000 grain weight increased by more than 23%, and grain yield per plant increased by more than 10%, despite a slight reduction in seed setting rate. So again, great achievement for rice varieties in order to get better yield. And the third example deals with barley, which is always problematic. So this is the gene GW2.1, this is grain width, and the gene in barley, also referred to has other names, was targeted in the study. It encodes E3 ubiquitin ligase, so it is involved in protein degradation and acts as a negative regulator of grain size. So if it acts as a negative regulator of grain size, people decided to try to knock out gene activity, looking for what will happen. So KO mutants in this gene using CRISPR-Cas9 led to elongated grains and increased protein contents, but also resulted in a reduction in yield with fewer grains per spike and lower overall seed production. So this is just an example of this trade-off between grain size and yield in the context of barley breeding. So not only they indeed increased grain lengths, what you can see here, for instance, this is nicely visible in comparison to wild-type plants. However, they had a reduction in yield because of fewer grains per spike and lower overall seed production. So now we will go to enhancing nutritional value of crops. So this is again the table, and where people published data on biofortification of plants through gene editing. And the examples marked by black arrows will be presented now in more details. So, enhancing nutritional value of crops, this study utilized CRISPR-Cas to achieve targeted insertion of a carotenoid biosynthesis cassette in rice, and it resulted in marker-free carotenoid-enriched rice varieties. So the engineered plants demonstrated high level of beta-carotene accumulation in seeds without any detectable morphological or yield parameters. And this work highlights the feasibility of using CRISPR-Cas technology for precise large DNA fragment insertions at genomic safe harbors, offering a promising approach for crop biofortification and genetic improvement. You can even see when you compare the seeds that the seeds of transgenic plants where biosynthetic pathway of carotenoids was inserted are more colored in comparison to wild-type plants. So the other example deals with tomato, and this is the only nowadays officially commercialized crop CRISPR-Cas9 product. This tomato, it is called Sicilian Rouge, high GABA, and it was introduced to Japanese market in 2021. And this tomato has been modified to contain higher levels of GABA, gamma-aminobutyric acid, which is beneficial for cardiovascular health. And it is available for sale and is commercially cultivated. And again, another one, this is for potato. So the researchers targeted SBE1 gene, it is a starch branching enzyme 1, and SBE2, starch branching enzyme 2, which are responsible for introducing branching points in amylopectin, a major starch component. And branching means the more branching you have, the better digestible the starch is. However, if you have less branching, and nowadays many people do not suffer from food shortage, but suffer from eating too much, and also in industry it is much more advantageous to have less branched starch. So they decided to inactivate both genes using CRISPR-Cas. So they generated mutants with reduced branching enzyme activity, altered starch granule morphology. So they increased amylose content and reduced branching frequency. So amylopectin was reduced. And novel starch phenotypes, such as tiny granules or granules with multiple HeLa. HeLa means the centers around which starch grains start to build. Yeah, these are HeLa. This is called HeLa. This study demonstrated the potential to create potatoes with modified starch properties, suitable for industrial applications, or resistant starch for health benefits without compromising other agronomic traits. Okay, so now we will come to developing climate-resilient crops. I skipped all slides I had about, climate, how it affects our life, and so on, because this is obvious for everybody today. So I just focus on the examples of plants with increased abiotic stress tolerance. And here you have also a table with two marked examples that I will describe more in detail. So the first one deals with, sorry, this is in rice. So this is the work was entitled CRISPR-Cas9 and CRISPR-CPF1 mediated targeting of stomatal development gene, EPFL9, in rice. So what is this CRISPR-CPF1? This is Cas enzyme derived from another bacteria. It is much more simple. This is single subunit, smaller, and is much more tolerant in the case of PAM sequence, and is more precise than Cas9. So people use more and more of this CPF1 in crop plants. So they used this CRISPR-Cas9 and CPF1 to edit this epidermal patterning factor like 9 in the rice, which regulates stomatal development. And the resulting mutant plants had an 8-fold reduction in stomatal density, which can improve water use efficiency and potentially enhance crop resilience to drought. So importantly, the gene edits were stable across generations, and the mutants were transgene-free, making them more viable for agriculture applications. The authors do not mention anything about yield, because you have to know that if you strongly reduce the amount of stomata, the gas exchange will be reduced as well, and transpiration will be reduced as well. So transpiration, fine. You get plant resistant to water scarcity. However, the efficiency and of yield could suffer quite a lot because of these stomata so strong number reduction. So this is better to manipulate, to rather reduce, but not to as much the number of stomata per plant to avoid the drop of photosynthetic efficiency. So the next one is also example concerning rice, and it was against salinity. And the study used CRISPR-Cas9 to edit the RR22 gene. This is response regulator 22 in rice. It functions as negative regulator to salt resistance. So again, if you have negative regulator, it is advantageous, but this is just you inactivate the gene. So KO, so knockout plants for this gene, should significantly enhance the plant's salinity tolerance without affecting its major agronomic traits under normal field conditions. And the authors obtained 2 homozygous mutant lines that showed improved growth metrics, such as shoot, fresh, and dry weights under saline conditions compared to wild-type plants. And you can see here at the picture, these are 2 transgenic lines and wild-type plant grown in control conditions, while here in salinity conditions, and both mutants with knockout gene of this RR22 grow much better in these conditions. So now we will switch to creating disease and pest-resistant crops. And here I should say that genome editing particularly is a useful tool and has revolutionized the development of disease and pest-resistant crops. And this approach accelerates breeding, precise processes, reduce the need of chemical pesticides, and so on. So now what about examples? So there are many examples. I will show just 2 that are marked by black arrow. And the first one deals with barley again. So this is a study that utilized CRISPR-Cas9 to generate knockout mutants at the locus encoding the gene microhidia1, MORC1, and gene in barley is associated with immunity, encoding chromatin remodeling ATPase. So from gene association studies, the gene was selected as the one that is associated with immunity. So the resulting mutants exhibited enhanced resistance to fungal pathogens, such as Bloomeria graminis and Fusarium graminerum, compared to wild-type plants. So this highlights the potential of genome editing to improve disease resistance and reduce reliance on chemical pesticides in agriculture. And the second example deals with grape wine. And in that case, a gene called MLO3, this is mildew resistance locus 3, was targeted by CRISPR-Cas. And again, knockout plants for this gene were obtained to enhance resistance to powdery mildew caused by Ericife necator. And the mutations introduced in MLO3 disrupted its function, leading to increased host cell death, cell wall apposition, and hydrogen peroxide accumulation, which collectively improved resistance. So among the edited lines, 4 displayed enhanced disease resistance, demonstrating that CRISPR-Cas9 mediated gene editing can be an effective strategy to improving fungal resistance in grape wines. What is important? So when you see here wild type and mutant, and when you look for hyphae, so you can see that in the mutants, hyphae are not developing, while in the wild type plant, hyphae are present. And if you measure total lengths of hyphae, you can see that in wild type it is quite a lot, while it is dramatically reduced in at least these 2 shown transgenic lines, CRISPR-Cas edited lines for MLO3. So now we will switch to ethics and regulations in plant genome editing, which is also an important issue. So there are, in some countries, regulations and differences made between GMOs and gene-edited crops that I will mention now. Then we will talk about regulatory frameworks, how CRISPR crops are regulated globally, like United States, EU, or Asia. So what is the definition of GMO and gene-edited plants? GMOs are genetically modified organisms, what means it involves insertion of foreign DNA into an organism genome, often using transgenic methods. While gene-edited crops modifies genome without introducing foreign DNA, typically by editing existing sequencing using tools like CRISPR. And this differentiation between gene-edited crops and GMO allowed many countries to allow propagation of gene-edited plants as for the market, while unfortunately in EU it is still considered to be GMO. Also, we as scientists and also many agricultural corporations fight to convince European authorities to change the law. And it is at its way, because I think nowadays European Union is the only region in the world that still does not allow to generate varieties, crop varieties using CRISPR-Cas. What resulted that we have the research concerning CRISPR-edited crop plants is really not really developing, because if people see no chance to sell their varieties, to propagate them, then the research also stopped. So in the United States, the US does not classify gene-edited crops as GMOs, and both USDA and FDA allow for production and being on the market of genome-edited plants. And US is a leader nowadays in gene editing research and commercialization with gene-edited crop like known browning mushrooms, and CRISPR-edited soybeans already on the market. So I think these non-browning mushrooms are quite famous, and soybean as well. And now what about China? So in China also gene-edited crops are not regulated as GMOs. And in 22, China introduced streamlined guidance to approving gene-edited crops, aiming to encourage innovation in agriculture. However, there still exists a public skepticism for acceptance of gene-edited crops. So in Japan, gene-edited crops are not regulated as GMOs, and Japanese government actively supports research in gene editing for food and agriculture, as this first example of tomato that was allowed to be sold on the market in Japan. Then South Korea, also similar to Japan, gene-edited crops are not considered to be GMOs as long as no foreign DNA is present. And South Korea actively promotes CRISPR-based innovations in agriculture. And in India, you are lucky because your government also allowed for gene-edited crops to be on the market, and they are not regarded as GMOs. And in India, as we learned looking in the internet, you have this famous flagship program in genome editing in plants. So genome editing for enhanced yield and quality traits, GE plants, project led by National Botanical Research Institute that aims to develop tomato, cotton, chickpea, rice, and mustard varieties with improved traits using genome editing technologies. And it started in 22 when the Indian Patent Office granted patent for ERS genomics, covering methods and compositions of using CRISPR-Cas9 in various cell types, including eukaryotic cells. And that paved the way for the legal use of CRISPR-Cas9 in scientific research in India, as well as agronomic. So now we will talk a little bit about future directions in plant gene editing and challenges and opportunities. So future directions in plant gene editing could be like genome editing of underutilized or orphan crops for food security, and integrating artificial intelligence and machine learning for gene target identification, and synthetic biology, beyond editing, creating entirely new pathways in plants. So we will start with underutilized and orphan crops. And these underutilized crops, what are these? These are crops grown on a small scale or in specific regions, often overlooked in global agriculture, despite their adaptability to challenging environments. So this could be like sorghum that grows in Africa, India, and China, fonio that grows in Mali, Burkina Faso, Ivory Coast, Nigeria, Senegal, so generally Africa, millet that grows in China, Japan, Bhutan, RPA. Does it grow also in India, millet? Yes, I think so, yeah. And amaranth that is Indonesia, Malaysia, India, for chow lai and harif sopu, right? Do you see it? These dishes? Yeah, okay. Africa, Greece, Americas. Okay. So orphan crops are crops that receive little attention in terms of research, breeding, and development compared to staple crops like wheat, rice, or maize. And here, as examples, we can mention teff, that grows in Ethiopia and Eritrea, cassava, South America, yam, never saw this plant. So it is in South Africa, Asia, Africa. Do you have it, this yam? You have it? Okay, and breadfruit. that is growing in Central America, Northern South America, and Caribbean. So these are these examples of orphan plants where do not get enough attention to be studied. So why focus on these crops? For food security, for sure, because many underutilized and orphan crops are highly nutritious and can grow in marginal soils, making them critical for regions facing food shortages. Then climate adaptations. These crops are often naturally resistant to drought, heat, or pests, and can survive where staple crops struggle. And the third is the diversifications of diet. So reliance on a few staple crops like rice, wheat, maize makes global food systems vulnerable to pests, diseases, and climate change, and enhancing orphan crops and diversified diets and reduce agricultural risks will be advantageous. So, first, we will talk about underutilized orphan crops for food security in improving nutritional content and enhancing yield resistance. The first, there are not so many studies, I have to admit. TEF. In TEF, you have genetic tools to manipulate CRISPR-Cas9 approach and was used to create knockout mutants in SD1 gene. This is semi-dwarf 1 gene, resulting in semi-dwarf plants with improved lodging resistance. This is important, a major issue limiting TEF productivity. So you can see here the wild-type plants and different TEF mutants that are semi-dwarf and have improved lodging resistance. Then sorghum. Sorghum protein quality improvement was done using CRISPR-Cas9 approach. In sorghum, the caffeurins are the major storage proteins. And in sorghum grains, and form protein bodies with poor digestibility. And caffeurins are devoid of essential amino acids like lysine, and they also impart poor protein quality to the kernel. So what was done, CRISPR-Cas9 was used to target K1C gene to create variants with reduced caffeirin levels and improved protein quality and digestibility. And what was obtained that T1 and T2 seeds showed reduced alpha-caffeirin levels, and selected T2 plants showed significantly increased grain protein digestibility and lysine content. So these are wild type, while these are transgenic seeds with improved protein content in sorghum. Then impact on food security, the regional benefits. It supports communities in Africa, in Asia, and in Latin America, and rely on these crops. It promotes self-sufficiency and reduces dependence on imported staples. And for global benefits, it reduces pressure on staple crops and contributes to sustainable agricultural practices by utilizing diverse crop systems. So now I will switch to integrating AI and machine learning for gene target identification. So AI is revolutionizing genome editing by enhancing the design, precision, and efficiency of gene editing tools, such like CRISPR-Cas, but TALENs and ZFNs as well. So advances like AlphaFold, the well-known for you program to design tertiary structures of proteins, protein-protein interactions as well, may be beneficial in improving genome editing enzymes and optimizing base editors for targeted modifications. And AI assisted by large language models, trained models. So we think about machine learning, like OpenCRISPR-1, are paving the way for the de novo design of genome editors, accelerating the creation of highly performance tools for crop improvement. And then these innovations are driving more precise genetic modifications, reduced breeding cycles, and enhanced crop resilience, making AI a crucial component of modern agriculture biotechnology. Not only, everywhere. So we can expect that AI, together with machine learning, will help us in designing better genome editors to use to edit crop plants. And now, at the end, synthetic biology. So beyond genome editing, creating entirely new pathways. So by leveraging synthetic biology, scientists go beyond modifying existing plant traits and instead to create entirely new functionalities. So, for instance, here at on the picture, you have synthetic rubber production in dandelion. So this is Taraxacum coxogis. We have Taraxacum in Poland, but officinalis, this species is not present in Poland, at least. And this species of Taraxacum was engineered to enhance rubber biosynthesis, providing a sustainable alternative to petroleum-based rubber. And the second example is even more promising because this is a way how to omit some natural negative feedbacks, allowing the assimilation of nitrogen by some bacteria. So how it was done? A synthetic rhizopine biosynthesis pathway was introduced into barley, enabling the plant roots to exude this rhizopine. This is called silloinosamine. This is a signaling molecule. And this was accomplished by integrating codon-optimized bacterial genes, 2 genes, into barley genome under strong plant promoters. And I think you are aware that codon context is different in different organisms. So we have more than one codon for different amino acids, and they are used with different frequency in different species. So bacterial codons have to be optimized to barley codon context. And from the other side, there is a diazotrophic bacterium, Azorhizobium collidans, that was genetically modified with a rhizopine receiver plasmid. So rhizopine was exuded by barley roots, and Azobacterium was modified, introducing a plasmid containing rhizopine receiver. So this plasmid contained also 2 genes for rhizopine sensing. And this synthetic circuit enabled rhizopine-dependent expression of NIF-A gene in Azorhizobium, the master regulator of nitrogenase activity. And nitrogenase activity is responsible for nitrogen fixation. So bacteria colonizing the rhizosphere of engineered barley responded to rhizopine by activating NIF-A, which triggered nitrogenase activity for nitrogen fixation. And this modified version of NIF-A was used to bypass natural feedback inhibition, allowing nitrogenase to function even in suboptimal conditions. So this is really, I would say, a dream from 80s of the last century are coming true, that we are now trying to be able to fix nitrogen artificially by modifying organisms, others than legumes. Yeah. So now, key takeaways messages. So gene editing tools like CRISPR-Cas9 enable the precise improvement of crop yields. Targeted modifications can enhance the macronutrient content of crops. Gene editing provides tools to develop crops that survive under extreme weather conditions. Edited crops with improved resistance to pests and pathogens reduce dependency on chemical pesticides. And exploring synthetic biology and multiplex genome editing can push the boundaries of innovation in plant breeding. And with that, I would like to thank you for your attention. If you have questions, I'm eager to answer. Thank you.